Hi there, my name is Dalton Miller, student ID number 1522. So today we're going to be looking at an example where we use radiation therapy to induce cell apoptosis in a mouse tumor model. So recently, radiation has been used to induce apoptosis so the cell can then die and spread is reduced. But we see that there's a trigger mutation in a PTEN tumor suppressing gene that causes the protein product to not act as a phosphatase to dephosphorylate PIP3 into PIP2, which then blocks against apoptosis and allows the cell to continue to function and progress and proliferate. So first we're going to look at why was this working before? Expression of cytochrome C. So looking at the pathway to see how apoptosis was activated, we know that signaling molecules are needed to institute this downstream effect. So a signaling molecule will come in contact with a G protein located in the transmembrane, and this transmembrane protein will become activated. This is GCPR. So as GCPR becomes activated, it can recruit other G proteins, such as alpha G protein, and this protein is going to be bound to GTP. This helps activate it so that as it is bound to the membrane, it can move and further recruit phospholipase C beta. So as it moves towards phospholipase C beta, phospholipase C beta becomes activated, and targets a transmembrane protein PR45 biphosphate, or PIP2. This is bound to different phosphate groups activated phospholipase C beta cleaves the phosphate groups from the PIP2, making DAG in the transmembrane and removing inositol 145 triphosphate, IP3. IP3. Now, IP3 has its own function. It's going to target a calcium channel release gate that is located in the lumen of the endoplasmic reticulum. So as this moves around and finds its target in the lumen, it binds to certain channels right here on the side, which open up the calcium channels. Once the calcium channels are open, it's going to release tons of calcium ions. We'll just say these are calcium ions, the scribbles. As calcium ions are released in high amounts, DAG recruits protein kinase C. So, as it recruits protein kinase C, it waits to be activated by, you know what? Calcium ions that were released by the IP3 binding to the calcium release gate. So as these ions move, they move, they move, they move, they move all the way until they bind into the activated protease kinase C. Protein kinase C. This protein kinase C will then trigger downstream activation to turn on specific genes used for regulation. This is the main pathway that we see as inducing apoptosis. PKC can move and activate intermembrane proteins that are located in the cell or in the mitochondria. And when it activates these certain proteins, such as BH123 protein, this triggers an apoptotic stimulus. Now, what is this apoptotic stimulus? So as a bunch of different proteins, signals are within the lumen, some organelle here, different proteins. These are each different proteins. One protein in particular is cytochrome C. So we know that cytochrome C is aggressively released when an apoptotic stimulus is felt. So as PKC comes activated by the calcium channels in the previous pathway, this will come and activate a protein in the transmembrane. This could be a BH123 protein. This protein has specific signals within the gene that tell the cell whether it wants to induce or restrict apoptosis. So as PKC comes and binds to this inactive protein, it will activate it. As this becomes activated, it will trigger opening of gates. So the gates open, and all of these proteins inside will begin to move out, until they are in the cytosol. And once these proteins are in the cytosol, cytochrome C specifically, the cell will recognize that it needs to undergo apoptosis, and it will begin to degrade and go into scheduled cell death. This is most likely the pathway we see that induces cellular death with radiation. In our example here. So FGF is overexpressed. We could say FGF could be the key protein that actually activates pathways to release cytochrome C. Now, the mutation in a specific gene, which is a tumor suppressing gene, PTEN, is of significant importance because once this gene was mutated, we were no longer able to get apoptosis of a cell. So why is that? So we know that a key pathway that prevents apoptosis is the RTK pathway. and specifically with the focus on the IP2 PIP3 protein attached to the membrane. So the PTN mutation acts so that phosphatase cannot dephosphorylate PIP3 into PIP2. Now, with that, we see a bunch of downstream signals that change, leading to the inhibition of apoptosis. So let's begin that. So a signal is received. This signal can be received by a tyrosine kinase. So a dual signal comes in, recruits two proteins to come together to activate a specific signal. So as this signal is caught, it activates the tyrosine kinase. This is the signal, and these are the tyrosine kinase. Tyrosine kinase. So as tyrosine kinase becomes phosphorylated, since it is a kinase, it could then phosphorylate itself and trigger own activation, and then phosphorylate itself, dephosphorylate itself. So as it becomes fully activated, it recruits a protein called PI3 kinase, which can work similarly to the protein that's synthesized by the PTEN gene. This protein then binds to one of the phosphate groups on the tyrosine kinase, and becomes activated. The role of this PI3 kinase is to phosphorylate the transmembrane protein PIP2, talked about in the last pathway. So PIP2 has two phosphates. The kinase will activate and phosphorylate this PIP2 an additional time to convert it into PIP3. Now, the critical protein that is being inhibited from the PTN gene would dephosphorylate the PIP3 so that it could stay in PIP2 Our other pathway, RTK pathway, can continue as progressed, releasing calcium, activating PCK, turning on downstream signals, part of the membrane, to release cytochrome C so that we can trigger apoptosis. However, if PIP3 remains, this protein, transmembrane protein that's phosphorylated, can survive by recruitment of two key pH domains. This is PDK1 and AKT excuse my drawings. I am not Pablo Picasso. So, as these proteins come and bind to the PIP3, this activates both PDK1 and AKT. So, DAG bound, another DAG bound. We'll say this one is AKT and this is PDK1. PDK1 becomes phosphorylated and activated. AKT becomes activated by the PDK1 to institute a phosphate group, and another motor protein, mTOR, comes and is already activated. mTOR will come and activate AKT as well. Now AKT is fully activated, so it's ready to regulate downstream signals. Once it's fully activated, the AKT will break off from the PIP3 in the transmembrane. Still maintaining its activation though. Now, as it's activated, it will move towards a protein called BAD. Bad. This protein is going to join active AKT. But it will be bound to another inactive apoptosis inhibitory protein right here. So this is the inhibitory protein. Used to halt apoptosis signaling. But it will not work if it is attached to the bad protein. So this is where active AKT comes in. So active AKT will recruit itself to the bad protein. It will phosphorylate the bad protein so that bad moves out is phosphorylated, and when it's phosphorylated, it recruits another protein called 1433, and this blocks off the bad protein so that it cannot activate or bind any other substrate. That leaves us with the inhibitory protein. So as the inhibitory protein moves, it will eventually find downstream signals that it will block. And this will trigger apoptosis to halt. Now, where could it block? Well, let's go back to the organelle. We know that PKC activates these transmembrane proteins, but we also know a reason for inactivation is the binding of a specific protein to transmembrane proteins that allow transport in and out of a cell. So, if our protein comes here, this is the inhibitory protein. It will block these pathways. If these pathways are blocked, then PKC cannot activate these pathways to open up. And if these pathways do not open up, then we will not see any transportation of molecules into the cytosol to tell the cell it needs to undergo apoptosis, especially cytochrome C. So if cytochrome C is maintained from within, we will see a resistance to apoptosis, and continuous growth. And these are some of the main reasons for why the cell with a mutant gene led to resistance to radiation to stop proliferation of cells that are undergoing tumors. Thank you. My name is Dalton Miller, and I hope you learned something today.